Professor Mike Keller, Head of the School of Agriculture, Food and Wine, Director of the Waite Research Institute and Dean of the Waite campus.
I'm an entomologist, and I've spent the last 40 years looking for ways to protect our crops from insect pests. I've worked on a range of different crops, ranging from corn, citrus, potatoes, broccoli, soybean. And in that time, I've noticed, as have many of my colleagues, that we see the same pests over and over again. In my case, I've seen a corn earworm feeding on corn. I've seen Citrophilus mealybug feeding on citrus, and cabbage aphid feeding on its host cabbage. It's not surprising that we see insects like this showing close associations with plants and only attacking a few of them. Plants have been defending against attack for as long as they've been around. And if we consider that plants have been attacked, they've evolved a range of defenses. In some cases, those defenses are physical. We see spines and hardened bark that work very effectively against insects and other animals. But it's chemistry which is the main line of a defense that they have. Put together, the chemical and physical defenses that a plant has against attack is like a great security wall that protects them from attack from pests. And it's only when a pest or a pathogen develops a range of different methods to overcome those defenses that it can ultimately be successful in attacking a plant. It's as if they found the key to unlock the door to get inside the plant and cause damage. Now, we're all familiar with locks and keys. What's the first thing you do if your house is broken into? Well, most of us would go about changing the locks on the doors and windows. And this gives us some comfort knowing that our security has been regained. Now imagine that this lock is a lock which is preventing attack from pathogens and insects on a plant. If by some chance a pathogen or insect gets the right key, then it can unlock the defense, gain entry, and cause damage. Now, just as that we have with our houses, what we'd like to do is to change the lock. So if we have a different lock, the key doesn't work. And so what I'd like to talk to you today is about an exciting new technique called genome editing. Genome editing is a way of changing the locks on plants and very neatly providing new security. In order to discuss genome editing, I needed to describe DNA. The DNA that we all have is a set of instructions. It's much like an instruction manual that defines how an organism grows and develops and it functions from day to day. DNA is made up of a series of chemicals that can be characterized by letters A, C, G, and T. Just as letters make words, and words make sentences, sentences make stories, so too the letters in a genome, or the plant's DNA, make up its genes. Now, with genome editing, we have a capacity to edit the genes that a plant has. So with a plant, the DNA defines the color of its leaves, the shape of its leaves, how big the shoots are, its roots, and importantly, its defenses. So with genome editing, we can edit the sequence of DNA much the same way that a newspaper editor can edit the text of an article. And genome editing goes by a range of different names like CRISPR-Cas, tail ends, and zinc fingers. Here at the Waite campus, some of my colleagues are using genome editing to explore the biology of plants like wheat and barley. But today I'd like to describe for you an example that comes from Iowa State University, where Ting Li and his colleagues used genome editing on a very important staple crop, rice. Now, rice is attacked by a devastating disease called bacterial blight. As you can see in this picture, bacterial blight can be devastating. It can destroy whole crops of rice. And the way that the bacteria does that is it switches on a gene called OS sweet. OS sweet releases sugar into a cell, and the bacteria diverts that sugar to satisfy its nutritional needs. What it does is it drains the cells of vital energy and ultimately kills them. The OS sweet gene itself is 2,883 base pairs or letters long. What I've done is I've taken a string of beads And I've precisely copied the sequence of the gene in this. So each bead, in this case, each colored bead corresponds to one of the letters in the DNA sequence of the OS suite gene. Now, Tingley and colleagues, what they did was they recognized that the bacterial blight pathogen switches on a promoter sequence. This promoter sequence is like a light switch. It's a series of DNA molecules that turns on the gene or turns it off during normal plant development. And so they recognized that they could somehow affect the way that the bacteria uses this OS suite gene by changing the promoter sequence. And you can see just how long the sequence of a gene like OS suite is, by how long it's taking me to take it out of my container. And right here at the beginning is the promoter sequence. What Tingley and colleagues did was they deleted 9 base pairs from this sequence. 9 base pairs, and when they did that, the result was wonderful. The plants that had the edited gene could resist attack from bacterial blight, and the plants that could be grown in the field without any damage caused by this devastating pathogen. We need to put this whole example into perspective. Tingley deleted 9 letters from the genome of rice compared to the 2,883 base pairs that were in that sequence of beads that I just showed you. But to really put it in perspective, you have to realize that the rice genome itself has more than 374 million base pairs. If I wanted to replicate the genome of rice in beads, I would have to make a string that would stretch between Rome and Moscow, or between Melbourne and Cairns, in order to represent the genome of rice. 9 base pairs were deleted. Put it another way, if we make the analogy between letters and the genome, the letters in a book, you would have to read the entire 7 volumes of the Harry Potter series more than 77 times in order to see the same number of letters as occurs in the rice genome. In comparison, the 9 base pairs that were deleted from the promoter sequence are equal in number to J.K. Rowling's name. What we have here is an example where tiny little change has made a very profound effect on the health of plants. Now you might wonder, well, they made that change, but did it affect the rice plants in some other way? Well, Tingley went and checked and found that the OS suite gene still expressed itself the way it should do naturally within the plant. So in this case, the lock was changed, and behind the defensive gate that was put in place, the rice plant still effectively continued to function. Now, before we rush to take on genome editing, we need to look at some of the alternatives that might be applied. One of the things we could do is spray pesticides. But pesticides aren't necessarily the best way to deal with pathogens like this. The pathogen will still cause some damage. Organisms other than the pathogen are going to be affected by that spray, and resistance to the spray can often develop. Alternatively, we could use genome editing to prevent attack by bacterial blight and from occurring at all. Another approach we could take is plant breeding. With conventional plant breeding, we would take various varieties of rice, cross the plants, and look to see if there was rare examples where resistance to the pathogen emerged. This can take a very long time, many years, and success is not guaranteed. That's one of the reasons why pathogens like bacterial blight are still a problem, because after many years, people haven't been able to generate the resistance that's needed. Alternatively, we can use genome editing to change the locks and ultimately protect the plants from attack by disease. Yet another way we could approach this would be through mutagenesis breeding. Scientists long ago realized that no matter how long and hard they search for resistance or other genes, sometimes plants just don't have the genes that they want to see in them. And so they've created random mutations and looked to see if they could generate what they need. Now, this is a routine practice in plant breeding today. Since 1930, more than 3,000 varieties of crops have been released and are grown around the world that are the product of mutagenesis breeding. And the way it works is that you use either radiation or some harsh chemicals to cause random mutations. And then, just like in conventional plant breeding, the seeds are planted out, many of them are planted out, and you go to look to see if there's a rare example where we see resistance to the pathogen. Success isn't guaranteed, and if we do find resistance, then we can say that in most cases, in fact all cases, we're going to see other random mutations occurring in those plants that come along with the resistance. And so we don't even know what those changes are. Or alternatively, we could use genome editing to very precisely modify the genes and achieve the desired result precisely and safely. Another way that we could approach this would be to study other plant species. Most plant species are not susceptible to bacterial blight or vites. So we could look at other plants that have natural resistance and look to see how it is that they succeed in resisting that pathogen. If we could find the genes that are responsible for that, we could transfer them into rice and ultimately achieve our goal. In principle, we could do this quite easily and effectively. But there's issues with transgenic plants. Around the world, many people don't accept transgenic plants. There's problems with acceptance. And even if we do this, it's a very, very expensive process to get it through the regulatory approvals. Or alternatively, what we could do is use genome editing to manipulate in small ways the genes that naturally occur in a rice plant. Now, it's important to realize that genome editing is a natural process. Plant cells, indeed all cells, have mechanisms where they snip and insert small amounts of DNA into their genome when the genes are damaged. And what scientists do is they harness this natural repair mechanism to effect genome editing. It's also important to note that other scientists can check. We can validate what's been done by looking at the changed gene sequences and check to make sure that appropriate and safe changes have been made to the plant before we release the varieties. After a long career working as a plant protection specialist, I'm really looking forward to the near future when we can more widely use the technique of genome editing to impart resistance to pathogens and insects that attack our crops. This is really going to improve the security of our food supply. Likewise, here at the Waite, my colleagues are working on barley and wheat and looking for ways that we can see those plants resist drought and salinity. So I think you should be able to see from today that genome editing is a precise and efficient and safe way to breed crops, and really looking forward to unlocking its potential as we move forward in the future.